Welcome to today's episode of 5-Minute Science Facts. Today we have a special guest here with us, Michael, who is going to tell us all about transcription and translation and why it's so important for us to work. So tell me, how does this work? What is transcription and translation, and why should people care about it?
DNA is the basic information unit of all life. DNA is copied into a complement known as mRNA or messenger RNA. This process is known as transcription, and it occurs in the nucleus of the cell. Transcription factors help regulate transcription and bind to the DNA to begin transcription. When transcription begins, the transcription factors stay behind while the mRNA polymerase begins transcribing. RNA base pairs come in and use the DNA as a template to make mRNA. After transcription has ended, the mRNA is exported out of the nucleus through the pores into the cytoplasm of the cell.
So that's all of transcription? It's really that simple?
Things can get pretty complicated with these processes, so I wanted to keep it simple for you. There are other factors that play roles in how transcription occurs too, but we'll save that for another time.
So what happens to this messenger RNA?
mRNA provides a message to produce a protein via the addition of amino acids to each other. This process is known as translation and occurs at the ribosomes in the cytosol of the cell. A ribosomal unit binds to the mRNA and recruits tRNA to bring amino acids to the ribosome. A 3-letter code in the mRNA corresponds to what amino acid is brought to the ribosome. Translation ends when a specific sequence is read by the ribosome.
So why is all this important in humans? The process is really cool, but what does it do for you and I?
Without transcription and translation, we wouldn't be able to function. From the time that we are just one cell, transcription and translation are essential for development to work correctly. During development, it is important that genes are active and expressed correctly throughout the organism. This is regulated by transcription factors, which control the amount of transcription in certain cells. Different cells do different things. For example, the cells of the brain do different things than the cells of the skin. You wouldn't want cells that are supposed to work in your skin to work in your liver. In order to see which genes are expressed in what place in the body, scientists use reporters, which help visualize the gene during development in the organism.
So what's an example of these reporters?
An example of this is a green fluorescent protein, which glows green when the protein is exposed to a particular wavelength of light. The top picture shows what a gene normally looks like, while the bottom shows GFP added into the DNA sequence. In order to add GFP into the sequence, the regulatory region is kept the same as the gene of interest, but the coding sequence is changed to that of the GFP DNA. GFP is attached to the gene of interest and transcribed and translated at the same time as the gene of interest. By using GFP, we can see if a certain gene is active at certain times or not. This shows a fruit fly developing with certain cell types tagged with GFP.
That's all the time we have for today. Thanks to Michael for telling us all about transcription and translation.